Now that you've watched sliding filament theory a few times, I'm hoping that you're getting a better hang of that information. What you're probably wondering, though, is how do those calcium ions get released? What is it that happens that causes the release of those calcium ions? And what it is that happens is an electrical signal is sent through a motor neuron. A motor neuron is just a cell in the nervous system that tells either a muscle or a gland to do something. The way that motor neurons communicate is by sending electrical signals down their axons, and then those electrical signals get changed into chemical signals to go from the motor neuron to the cell that it is affecting. So you'll hear me say either action potential or impulse. Both of those are the same thing. Both of those are those electrical signals that can be transmitted along a cell. So that motor neuron is going to send its electrical message down its axon, and eventually it's going to hit the end of the axon, or the axon terminal. When that action potential reaches the axon terminal, it's going to open voltage-gated channels that line the axon terminal. If you remember from your general bio class, voltage-gated channels are channels that allow specific ions through, but only when they're opened by the appropriate voltage. And so here we're working with calcium ions, and those voltage-gated channels are going to open and allow calcium ions to move into this axon terminal. The calcium ions are going to trigger the release of a neurotransmitter. Here we're talking about acetylcholine. So acetylcholine is stored at the axon terminal in synaptic vesicles. And vesicles are just little bags of cell membrane that hold products that the cell doesn't want floating around everywhere. So here we have little bags of acetylcholine. And when the calcium goes rushing in, those bags of acetylcholine are going to float down to the cell membrane and release their contents into the area between the axon terminal and the muscle cell that it is affecting. So that acetylcholine is going to travel in this area, and this area is called the synaptic cleft. It's the space at the synapse between the motor neuron and the muscle cell. And eventually it's going to bind to proteins on this side of the synapse, and those proteins are going to trigger a change inside the cell when acetylcholine binds to them. So acetylcholine's released, they bind to a protein over here, and that triggers a change inside the cell, and it actually triggers an action potential to travel down the muscle cell. That action potential is going to follow kind of the external edge of the muscle cell, but it's also going to go down these T-tubules that you learned about last week and bring that signal deep into the muscle fiber. That signal is going to trigger the terminal cisternae to release calcium into the cytosol, and that calcium is going to bind to troponin and cause that whole sliding filament interaction that you've been studying to occur. Okay, so just to do another quick overview, action potentials are nervous impulses. They are electrical signals that travel through a motor neuron. At the axon terminal, the end of the axon, that electrical signal is converted into a chemical signal by the release of acetylcholine into the synaptic cleft. Acetylcholine is going to bind to proteins on the motor end plate. The motor end plate is part of the sarcolemma that has a lot of infoldings. That those proteins that are attached to the motor end plate are going to change the internal environment of the muscle fiber in a way that sends an action potential throughout the muscle fiber. That action potential is going to travel down T-tubules, triggering the release of calcium from the terminal cisternae. I hope this video helps to clarify this. Again, this is just kind of a preview to action potentials. We're going to study this in a lot of depth when we get to the nervous system. Right now, I really just want you to understand that it's an electrical signal that travels down the axon of a motor neuron. When it gets to the end of that axon, that electrical signal is going to convert into a chemical signal by the opening of those voltage-gated channels, the influx of calcium, which triggers the release of acetylcholine. Acetylcholine is going to go through the synaptic cleft. It's going to bind to proteins on the motor end plate. When it binds to proteins on the motor end plate, it actually is going to convert back into an electrical signal and action potential that travels down the muscle fiber, down into the muscle fiber through the T-tubules, triggering the release of calcium. And that calcium is the calcium that binds to troponin and allows for the filaments to interact with each other and contract the sarcomere.